Hello, my name is Jenny Crane. I am the Virginia Department of Health Office of Epidemiology, Genomics Program Manager. Our state is part of the Virginia Pathogen Genomic Centers of Excellence. This recording is a training presentation of a use case scenario for other states to use in their own jurisdictions. And this may be useful for either following along in a group setting in which you use multiple training materials and walk your own staff through some of the discussion questions, or you can use this to replicate the scenario on your own after having watched these slides. This presentation is entitled Genomic Epidemiology Result Interpretation, and it's specifically a healthcare-associated infection use case scenario regarding a MRSA NICU outbreak investigation. So to quickly review how this training scenario is designed, I will first run through the different handouts that will be available to you. Then I will talk about the background of MRSA. Next, we'll do an overview of the entire scenario and then break for discussion questions. So just to briefly show you what should be available to you, there's a single page handout that is a very brief description of the case study. It has instructions and then also references that may be helpful for understanding SNP thresholds or infection prevention and control recommendations for such an investigation. The second handout should be a 4-page document that goes through the specifics of each separate outbreak. And it also includes a SNP matrix and a phylogenetic tree for both outbreaks combined, as well as the bioinformatic methods that were followed by our state public health laboratory when they produced these genomic results, as well as some references about the software that were used when creating these visualizations. Then you should also have a single-paged de-identified epidemiologic line list that covers as many of the patient details that we could release about outbreak 1 and 2. Please reference this line list throughout the training. Then you should also have a single-paged handout that is an interactive training scenario worksheet. It's a 2-page document, and we will walk you through some of the steps and then the discussion questions that are included in this worksheet you would need to talk about on your own with your staff. And then there's also a instructor's guide with some possible suggestions or recommended answers to these questions. So these resources will be available to you with this training recording. We will also provide our contact information at the end of this training, and you can reach out to us should you have any questions about these handouts themselves. All right, so the overall goal for this training is to help staff apply genomic and epidemiologic information from a public health case study involving clusters of methicillin-resistant Staphylococcus aureus to learn more about laboratory result interpretation. So the overall objectives that we would like to achieve are to describe how genomics complemented the epidemiologic investigation, to report on the overall workflow of the investigation as well as the main findings, and then to identify how public health recommendations may be informed by different genomic outputs. So for just some background on MRSA to make sure everyone is all on the same page, MRSA is not a new pathogen. We've known about this as a major public health threat since 1961. It has one health relevance, and so MRSA can be isolated from animals as well as the environment. 30 to 70% of humans are carriers of Staphylococcus aureus, and about 80% of neonates are colonized with Staph aureus by 10 days of life. So some of these specific background information we have added to this presentation based on doing this use case scenario with live staff here in Virginia. And we got a lot of really specific questions about Staph aureus carriage. So hopefully these background information details will help you as you walk through this scenario. The major problem in terms of it being a public health threat is the fact that it is resistant to methicillin as well as other beta-lactam antibiotics. But when you think about MRSA and its ability to colonize infants, neonates can acquire Staph aureus as a part of their normal developing microbiota, yet this colonization predisposes them to invasive infection. And so it is estimated that this colonization can occur from a range of time from about 2 hours to 2 months after birth. And some of the references that we looked at said that neonates can acquire MRSA on average about 9 days after admission to a neonatal intensive care unit. So that specific environment puts them at a higher risk. And that acquisition can occur between 1 days up to 91 days after birth. Sorry, after admission to that NICU. So when you think about Staph aureus and prevalence, Staph aureus infection is estimated at 45 infections per 10,000 hospitalized infants. So about 1 in every 5 colonized neonate develop this infection. And the median onset time from MRSA colonization to infection can also range from an average of 4 to 9 days. So just think about this in context of the line list when you're looking at the details in this exercise. So this is a little bit of an older report from CDC, but is the most recent antibiotic resistant threat report that we could find specific to MRSA. So CDC estimates there are about 75,000 invasive infections of MRSA every year, and that results in about 10,000 deaths. This is about a 16% decrease, at least in 2019 from previous years. So these outbreaks occur in a multitude of settings that can include, healthcare associated, as this presentation is focused on, but not only hospitals, also long-term care facilities and other congregate settings such as daycares and correctional facilities. It's also important to remember that MRSA can be transmitted in community settings as well. So while this presentation is focused on healthcare, there could be community introductions into a healthcare facility as well. So some factors that make MRSA more easily transmissible has to do with what's called the 5 Cs. So crowding, frequent skin-to-skin contact, compromised skin, that can be cuts, abrasions, that can occur from various things: tattooing, body piercing, or injection drug use, contaminated items and surfaces, and lack of cleanliness. So just think about all those 5 Cs and where, which settings or locations they're most common in. So schools, dormitories, military barracks, athletic gyms, households, correctional facilities, and then specifically daycare centers or this presentation is obviously focused on those facilities that are licensed or regulated by departments of health. So just to keep in context that MRSA, is prevalent in other settings as well as what we are focusing on today. And then last thing I just want to mention, as we said in the first slide, that this is a zoonotic or One Health pathogen. It can also be transmitted in areas where animal handling is common, so veterinarian clinics and livestock settings. So just to bring this, MRSA background into the context of what we do in public health, MRSA is not a nationally notifiable disease condition. So there are no CSTE case definitions. And so when you're thinking about this specific outbreak and you're comparing it to what you're used to doing in terms of knowing a surveillance case definition versus establishing an outbreak specific case definition, MRSA is in a different category in and of itself. So, all outbreaks, 2 or more cases with an epidemiologic link, are reportable in most states. And so in most cases, your state may be doing an investigation involving an outbreak or a cluster of MRSA. But just in terms of how is this defined, what is the case definition, right? It's a little bit different than other pathogens we investigate routinely. I'm going to give you some Virginia specific stats here just to encourage you to make sure that you're aware of what these are in your own state. So individual cases of MRSA infections historically were lab reportable in Virginia up until about 2016. VRSA, or vancomycin resistant Staph aureus, is still on the lab reportable disease list. It is more rare, but in Virginia, we have the same disease control manual public health actions for VRSA as we do for MRSA. So when people come to our central office for recommendations or guidance specifically, a lot of that information is similar. So the other thing that's important to note for MRSA is that in Virginia, invasive MRSA infections, so single cases of that, were required to be reported by laboratory directors in Virginia from about 2007 up until that date of 2016. But beyond that, certain healthcare facilities are required to report MRSA laboratory identified events to the CDC's National Healthcare Safety Network, or the NHSN, as a part of federal inpatient quality reporting program. So these data are also shared with our Department of Health through NHSN data. So there are other ways to get some of this information even beyond 2026. But it's important to think about the context in terms of MRSA becoming more prevalent and why these changes have occurred over time in terms of individual cases no longer being reportable, and that we don't necessarily investigate every single case. On top of that, there are additional regulations in Virginia. We have a Board of Health regulations for licensure of hospitals in Virginia, and these hospital settings are required to report 2 or more epi-linked Staph aureus infections occurring in obstetric or nursery units to the local health department. So notice I said Staph aureus, so not necessarily MRSA infections, but any 2 Staph aureus infections in a NICU or in obstetric unit should be reported to the health department. So again, clusters. And then all outbreaks of MRSA are required to be reported to the health department in accordance with our Virginia code regulations. So again, I encourage you to research that for your own state, just to make sure that you're aware of what's reportable and what's not. The main thing I want to highlight here is the uniqueness of colonization when it comes to healthcare-associated infections, and specifically MRSA. So transient colonization or carriage is possible. Sometimes when we're looking at the actual laboratory results and we're trying to place this in the context of person, place, and time, and trying to understand transmission about individual cases, we sometimes forget that, carriage may be there, but colonization may be sort of come and go, depending on the individual case's immune system. So these next 2 bullets are as a part of our disease control manual as reminders for investigations that even if you have a positive culture or screening test, this doesn't necessarily indicate the presence of an infection, right? Unless there is a clinical correlation with signs and symptoms of the infection. And more importantly, I think this is where we forget, a negative culture or screening test from 1 or more multiple body sites is not confirmatory that the individual does not have any colonization with MRSA. Each sample or specimen that we take and then we test in the laboratory is, a single point in time in which that particular sample may not have just been good enough, in terms of the amount of biological material that was obtained from the sample site or the patient themselves. So when we are trying to tease out transmission, some of these facts are easily overlooked. We need to also remember with MRSA that transmission can be vertical or horizontal. And when we think about vertical, this is the mother to child transmission, that can occur vaginally or via the nose, the nares, or skin between the mother and the baby. Horizontal transmission is normally what we are thinking about investigating in these healthcare-associated infection outbreak scenarios, where it could be transmission from the nose or nares, as well as the skin of, say, healthcare workers from an infant to a patient. It could also be, a visitor or a family member in a NICU, for example, to the patient, as well as environmental exposure. We know that Staph aureus, as well as other healthcare-associated pathogens, can persist in the environment as well, and that is considered horizontal transmission. All right, so for the background on Staph aureus genomics, I'm not going to go into the weeds, and this image of the genome, while extremely detailed, is not the point here. I just want to have everyone have a little background on the basics in terms of Genomics 101 for MRSA. So this is a 2.8 million base paired genome for this pathogen, and about 75% of that genome is conserved. And it's conserved across all Staph aureus strains. So this is important to remember that, there is 25% variation between strains, and it's that variation that has contributed to over 5,000 distinct sequence types of Staph aureus. And it's these sequence types, or these genetic differences, that we use to track clones and to understand transmission, as well as population structures, or what strains are persisting in communities or hospital settings, for example. So So how does this genetic divergence or differences, how does this diversity, how is it acquired? You are likely familiar with mobile genetic elements as genomic epidemiologists, but for review for others of you that may be attending this training or doing this particular scenario, it's important to understand that these mobile genetic elements are often what confer antibiotic resistance. So for MRSA, it is this MECA gene that has been identified to carry antibiotic resistance and also makes this pathogen more virulent. The first handout that I've given to you provides a little bit of published literature that describes the mutation rate of MRSA, which is spelled out for you here as 1 × 10⁻⁶ substitutions per base pair site per year. And so when we compare MRSA to other bacteria, this is relatively fast. They have a conservative SNP cutoff range of about 25 SNPs, and this conservative SNP cutoff is again from one of those publications, but this cutoff is above which transmission of MRSA can be assumed within the previous 6 months. So we use that SNP cutoff to rule out cases from being associated with this specific cluster, right? So WGS is often easier and more helpful with ruling out cases than ruling them in. And as you know, SNP cutoffs are typically a rule of thumb. What's most important is that we use these thresholds in conjunction with epidemiologic information that we have collected as a part of an outbreak investigation, and understanding, when those samples were collected, which patients they were collected from, and the different interactions or exposures of those patients to each other and to the environment or the setting in which the outbreak is occurring. So again, SNP thresholds are a rule of thumb guidance. You need to take them with a grain of salt, but confer with the references that I've provided for you as well. And this again is just some background information to give us a leveling of the playing field here. So again, MRSA readily picks up these advantageous genes through horizontal transfer, and this speeds up the evolution of the pathogen's antibiotic resistance and ability to persist in the environment and to transmit readily to other patients. These genomic rearrangements and gene acquisition continue to occur as the pathogen evolves. So what does this mean for public health? These are just main categories that we've kind of already gone through already, but we need to remember how the mutations affect their evolutionary change of each pathogen or each isolate over time. And it's this genetic divergence that we use for constructing phylogenetic trees and for understanding genetic similarities or differences as we are trying to understand disease transmission in clusters of patients. As we know, SNP thresholds are a rule of thumb, but if it's small SNP threshold, this indicates close epidemiologic linkage and potentially a most recent common ancestor or exposure. Genomic rearrangements, as we said, can signify genomic instability, and these typically occur in regions that encode resistance or virulence. All of these rearrangements obviously indicate adaptive evolution, but it can also indicate a strain's divergence from ancestors. This is important for tracking these virulence gene acquisition or loss. And as we have said, this horizontal gene transfer is a key mechanism for acquisition of AMR, virulence factors, and then it's also important to remember that this can also be transmitted between species on plasmids. And detection of these elements across unrelated isolates can imply common selective environments and not necessarily clonal spread. So all of these factors, when we think about genetic diversity, is important to remember when looking at the genomic results that you have available to you. All right, so that was a bit of an exhaustive background, but hopefully it was review for most of you, or if it was new information for some of your staff, hopefully that gave them enough information to apply to this outbreak scenario. So as I said, we will be focusing on a MRSA outbreak that specifically occurred in one hospital, but it was a multi-month investigation. And it was menacing because it happened over a period of time in which multiple investigations were required, multiple outbreaks were detected. And this is just our credit slide here. Many of our state laboratorians were involved with helping us generate these genomic results, as well as the investigators who did all this field work and were kind enough to share this information with us to create this training scenario for all of you. So thanks to them. All right, so just for the overview, our Virginia Department of Health was notified about a suspected MRSA outbreak in a neonatal intensive care unit at Hospital A in early May of 2022. So as I said, even though they fully investigated and deemed this outbreak to be closed by the end of the month, a new suspected MRSA outbreak was reported by the same facility in September of 2022. So WGS revealed that both of these outbreaks were caused by MRSA of the same sequence type 105. So for this scenario, your genomic epi team has requested a comparative genomic analysis of all of the sequence isolates from both outbreaks to evaluate the genetic relationship between isolates and to assess whether separate introduction events occurred at Hospital A. So the task for you today is to use all of the epidemiologic investigation data provided to you and the genomic results, apply what you know from this background, including the mutation rate of MRSA, as well as those SNP thresholds, and what you understand about MRSA transmission, the different types of transmission, and to think also about genetic diversity from those, the background of the Genomics 101, but also how diversity can occur between hosts or between humans, and also within the same host, right? So in many healthcare associated infections, we can have multiple samples from the same patient in different body sites. So you need to think about all of those things when you're analyzing this information that we're going to provide to you. All right, so just to review the two outbreaks and how the timeline and sequence of events occurred. On May 4th, 2022, the first case had onset of symptoms and a positive blood culture and grew from that same specimen. In response, the hospital collected colonization screening specimens from 4 other infants, and 3 of them were deemed to be positive with MRSA. Correction. So 4 were tested and 3 were colonized with MRSA. On May 6th, an additional infant developed symptoms, even though his colonization test the previous day did not detect MRSA. So for this patient, a blood sample that was collected on May 8th was positive via culture. An infection and prevention site visit was arranged on May 13th, which revealed that the hospital NICU was divided into 3 different pods of 5 isolates. And so isolates are those warmer bed cribs that are on wheels. So just remember they're mobile. This hospital NICU had 1 anteroom for hand washing and donning PPE, but only 1 main patient care area. Isolates are kept in pod groups because there were no doors or partitioning walls to create separation between the pods. Each pod has a separate nursing station for charting. 4 nurses were assigned at the NICU at the time of the site visit, and the census was a total of 10 infants. All 5 cases were in Pod A with 2 assigned nurses. Pod B had 1 infant and 1 nurse, and Pod C had 4 infants with 1 nurse. State laboratory was also contacted to request whole genome sequencing for 2 culture positive patients' isolates, and Hospital A was advised to forward the other patient swabs for confirmatory testing as well. WGS characterized the outbreak strain as MRSA sequence type 105. All right, so this just runs through the specific genomic results from that outbreak 1 at Hospital A. And as I said, there were 4 isolates from 4 patients that were sequenced. 3 were within 10 SNPs apart from each other. The 4th isolate was genetically distinct or greater than 10 SNPs from the other 3. Okay, so was the outbreak over or was it? The hospital initially reported 4 colonized infants and that the index case, known to have been previously colonized and placed under isolation precautions, was then admitted to the NICU via transfer from another hospital, B, on August 29th. So again, look at your line list and see where this particular case was. So for outbreak B, investigators believe this was the index case. In response to that, on September 5th, the hospital collected colonization screening specimens from all 11 infants that were in the NICU at that point. Testing revealed 8 additional infants that were colonized with MRSA, who were also then put on isolation precautions. Another IP site visit was scheduled by the health department on October 3rd, and the health department recommended forwarding PCR positive specimens to the state lab for confirmatory testing and WGS of any positive blood culture isolates. And again, for outbreak B, the MRSA sequence type was identified as 105. So this outbreak strain persisted. Total of 4 isolates from 2 patients were less than 10 SNPs, which indicates that the isolates appeared to be highly genetically related and therefore more likely to have come from a common source. 2 additional isolates from multiple cultures from a third patient were genetically distinct, or greater than 10 SNPs, from the cluster of the 4 isolates from the other 2 patients. So again, it's really helpful to look at your line list here, noting where it specifically has a number and a letter that designates more than one specimen from the same patient. Okay, so the activity instructions. Again, you were part of a multidisciplinary team at your Department of Public Health that has been called in to help with this investigation of MRSA in the healthcare setting. So your GenEpi team is to review the public health case study information, all of the genomic results that are available to you, as a result of requesting a comparative genomic analysis across both of the outbreaks, and then use all this information to address the discussion questions. All right, as I mentioned, you're given a SNP matrix, and so we have just annotated this with color to make it a heat map with just 2 different thresholds here, less than 10 SNPs or greater than 10 SNPs apart. And again, as I mentioned, the isolates have a letter and a number and a letter indicating that there are multiple isolates from patient 5 here, A, B, and C, and patient 6 has 2 isolates from the same patient as well. So pay attention to that when you get to the phylogenetic tree and you are evaluating genetic relatedness here. Colors indicate which isolates were associated with which outbreak based on the epidemiologic investigation. So outbreak 1 is green, and when you look at your line list, that will be also outbreak 1, patients that were associated with that investigation, and yellow will indicate the patients and isolates that were associated with the second outbreak investigation. Okay, so what we have done as a part of this comparative genomic analysis is take all of that information and pull it into a MicroReact project for easy visualization and interpretation. So if you're not familiar with MicroReact, you can go to microreact.org and read a little bit more about how this genomic epi visualization tool works on the back end to integrate the genomic results with the epidemiologic investigation metadata. And hopefully this exercise will help you understand how this relatively simple visualization can allow for different inferences to be drawn. So just to quickly walk through what these different views look like to help you understand, we have created one view that's specific to outbreak 1, one specific to outbreak 2, and this is done by literally just excluding different isolates from the metadata that you don't want displayed in the SNP matrix panel or the phylogenetic tree. And then the view that you're looking at right now is the comparative genomic analysis where outbreak 1 and outbreak 2 are included together. We also made a view for an outbreak network diagram, and this was done in microbe trace. And just to make sure that there's no confusion, what we did with microbe trace, which is a different genomic epi visualization software, is we made a network diagram and just took a static picture of that network diagram and pulled the image file into MicroReact for viewing alongside the other panels. So as you can see in this visualization, we have used the shape of the nodes to indicate either a location, and in this case, this is all within the same hospital A. We don't have hospital B depicted in this network diagram. But looking at your line list, you should be able to tell pod A patients, from pod B patients, from pod C patients. And if these patients moved between pods, you can also see that displayed here. So outbreak 1 is represented by nodes with a triangular shape. Outbreak 2 is denoted with nodes of a circle. And what we have done with the colors of the nodes, just to make sure it's clear, the locations are in red, pods A, B, and C, and patients are in blue. The link legend over here on the far right helps you understand which patients were associated with which pod. So in pod A, we have outbreak 1 patients represented with the triangle in blue, and outbreak 2 in circles. But all of these patients were located in the pod A at some point. So this is what the purple links are. Pod B links are in green, and so you can see here that patient 14 was transferred between pod A and pod B at some point during their hospitalization stay. And the orange link is for those patients that were associated with pod C. And so you can see patient 13 and 15 were transferred between pod A and pod C during their stay. All right, that was kind of an exhaustive review of what the network diagram is displaying here. It will be up to you to integrate this information with the SNP matrix, so the single nucleotide polymorphism difference between each of these isolates as you're comparing them 2 by 2, the phylogenetic tree, which is representing the genetic relatedness in terms of evolutionary divergence of these isolates taken from different patients over time, and all of the epi metadata that's available to you in the line list, which came from the epidemiologic investigation. So with that, we will stop here and allow you to pause and review these discussion questions on your own with your staff, as well as using the instructor guide of suggested answers and possible inferences that you can pull away from these visualizations to discuss as a team as you learn more about integrating genomic results with epidemiologic information for the application and use during epi investigation. Again, case study contributors are listed here. We had a lot of help from the actual investigators in Virginia that collected all this information, as well as our laboratory partners at the State Public Laboratory, the Department of General Services, Division of Consolidated Laboratory Services. So I wanted to just acknowledge them as a part of this effort in creating this use case scenario for use in other jurisdictions. Thank you for watching.